Today we're going to talk about some basic concepts of clinical microbiology. So we're going to talk about types of infectious organisms, sort of the spectrum of organisms that cause disease in human beings. We're going to talk about the process of infection, so the process in which an infection can move from one person to the next. We're going to talk about basic standard precautions and enhanced precautions for the control of infection, particularly as we see them in the hospital setting or in clinical settings. And we're going to also talk about the problem of antimicrobial resistance, where the drugs that we use to prevent infectious disease are actually becoming less and less effective because organisms are adapting to them. So we're going to talk about these issues, and I just wanted to explain a little bit about what you're looking at, this picture here, which I really am fond of. So what we're looking at is a petri dish, which is a plastic dish used in microbiology that is at the bottom of the dish we have this media which contains lots of nutrients and lots of food that microorganisms, particularly bacteria, can consume and grow. So a petri dish like this is where we culture a sample. So we put some sort of sample on a petri dish like this and we let it sit under controlled conditions and see what it grows. So what we have here, which is pretty obvious, is someone just put their hand on this petri dish and we just cultured whatever was on their hand. And this was, I assure you, what they thought was a clean hand. It may not be freshly washed, but it was a clean hand. And you can see that there's a wide spectrum of organisms growing, right? Lots of little colonies of different colored and different shaped organisms growing just from this sort of clean hand being placed on the culture media. And it really tells us a lot about microorganisms in our environment. We have a wide variety of microorganisms that are present on our body, on objects in our environment, and the majority of organisms are non-harmful to humans. The majority of microorganisms in our environment are non-harmful. There's actually only a small spectrum of microorganisms that are what we call pathogenic. And by pathogenic, I mean able to cause disease in human beings. And it's just this small spectrum of microorganisms that are actually the focus of clinical microbiology, meaning the application or understanding of these pathogenic microorganisms in the treatment of the diseases and the prevention of the diseases that they cause. So in this slide here, what we're looking at is a table where different classes of pathogenic microorganisms are laid out. Okay, so these are different classes, and then there's some descriptions of them. So we've got the relative size, so you can see that we're going from viruses, which are the very smallest infectious microorganisms, to helminths, which are a type of parasitic worm, which are actually quite large, right, up to 10 meters, right? They can grow very, very large. And then we've got the site of reproduction, so where these microorganisms live and reproduce themselves. And some of these microorganisms live inside of our cells, right? That's where they like to occupy, that's where they're going to reproduce. And the rest, most of them, occupy the extracellular environment. Okay? So they live in things like skin and mucous membranes and other locations. And then there are some examples of diseases that fit in these classes. Okay? So as you can see, there's a wide variety of classes of pathogenic microorganisms. Generally speaking, the classes that we see most commonly include viruses. Number one, they are the most common pathogenic organisms. Bacteria, right, which tend to be the second most common pathogenic microorganisms. And then to a lesser extent, but still more common than the rest, are fungi, right, fungal infections. So if we focus on those three for a moment and take a look at them, what we see here is some basic morphology or the physical structure of some of these microorganisms. So over here to the left, we've got different types of bacteria. So the bacteria that we are used to hearing in terms of disease-causing bacteria, the way bacteria are named are actually based on their physical structure. So bacteria are single-celled organisms. So they are single-cellular organisms that in their single-cell shape, they can have different basic shapes. So a spherical single-celled organism or single-celled bacteria we call a cocci. A rod-shaped or oblong-shaped single-celled bacteria we call the bacilli. And then the sort of almost worm-like shape we call a spirochete. Okay. Now, if you notice in the cocci, when these cocci grow in clusters, so when they divide and they grow in clusters, we actually call them, we call this form of reproduction, we call them a staphylococci. Okay, so these are spherical, single-shaped organisms that tend to grow in clusters. That is what a staphylococci is. Okay, and then in contrast to that, if you've ever heard the term a streptococci, these are spherical-shaped bacteria that tend to reproduce and form chains. Okay, so when you're looking at a sample, for example, cultured from someone who's ill, when they grow spherical-shaped cells that grow in clusters, we now know that this is a staphylococci, some form of staphylococci. And if they're forming in chains, we know this is some form of streptococci. Okay? So it's part of the identification of the bacteria. If we look over here to the top right, the B channel or B panel, we see a typical virus. Okay, so this is the shape of a typical virus. It's very, very small compared to the rest of the microorganisms. And viruses are non-cellular, so they are not composed of cells. In fact, a virus really challenges our idea of what a living organism is, because a virus is just genetic material. Okay, so at its core, it has some sort of genetic material. It can be a strand of DNA. It can be a strand of RNA. Okay, sometimes the RNA structure is double-stranded. Sometimes it's single-stranded. So it's got some sort of genetic material. And then surrounding the genetic material is a protein coat. Okay? And that protein coat can be quite elaborate, as you see in this picture, where there's both a protein coat and then a protein and glycoprotein capsule around it, okay, or envelope around it. But this virus, that's very simple in structure, must get inside of a host cell in order to reproduce itself. Okay, so viruses are really parasites in nature. They must infect a host cell and use that cell's protein manufacturing machinery, the cell's the contents of the cell, in order to reproduce itself. And now at the bottom here, what we see is a basic structure of a fungus, where a fungi are multicellular organisms that they are sort of a cross between a plant and an animal cell. So they're very interesting in their reproduction pattern. But fungal infections tend to infect the surface of the skin or the surface of some epithelial structure. And they will form both spores, right? And these spores detach from the original fungus and travel through the air or travel by contact, and that's how they infect and spread. And they also, fungal infections or fungus in general in nature, also produce a quote unquote fruit, which is what we observe as a mushroom. Okay? So in nature, when you see a mushroom, that is actually the fruit of some fungus. Okay. So viruses and bacteria are two important categories of microorganisms that cause disease in human beings. Now, with bacteria, when someone has a bacterial infection, we can go through some steps in order to identify the bacteria. The most important step is to culture the bacteria. So we take a sample, we take a sample, a throat sample or a sputum sample or whatever bodily secretion that we expect to see the bacteria, and we grow it in a petri dish. And then that way we can actually clearly identify what the bacteria is. Another part of bacterial identification is applying the Gram stain. And you may have done this at some point in some microbiology class that you've done previously. But the Gram stain is a very standard and very simple chemical reaction. It's sort of multiple steps. And it helped differentiate two major classes of bacteria, gram-positive bacteria and gram-negative bacteria. So very briefly, we've got our culture, we've got our sample of bacteria on this slide, and we're going to apply first a dye called crystal violet. Okay, so it's purple in color, and it's a dye called crystal violet. And as you can see, we've got a variety of bacteria here, some bacilli, some cocci, and they all get dyed purple in this first step. In the next step, we apply iodine. And what happens is the iodine attaches to the crystal violet and creates this very large structure for the dye. Okay? And that's a very important thing because for some of these bacteria, that large structure gets trapped inside the cells, and for some other of the bacteria, it's still able to get out. Okay, so then we form this crystal violet iodine superstructure. The third step is that we apply this alcohol wash. And what the alcohol does is for some of the bacteria, it removes that color. Okay? For some of the bacteria, it's going to wear away on the first layer of the cell wall, and it's going to allow that crystal violet iodine to escape. Okay? And so you can see the bacilli in this slide have been decolorized, but the cocci remain violet. Right? So then the last stage is that we apply what we call a counter stain, which allows us to see the ones that were susceptible to the alcohol, okay? And it turns them pink. So now, after that final step with the application of the counter stain, what we clearly see are two colors that these bacteria take on. The original crystal violet and then this saffronin pink. Okay? And the saffronin pink bacteria are what we call gram-negative, and the ones that remain crystal violet are gram-positive. So we now have identified two different kinds of bacteria. Now, what does gram-positive and gram-negative mean, and how is it significant in terms of clinical microbiology, in terms of disease?